So good afternoon, everyone. It's my pleasure today to introduce our next webinar held by Professor Wolfgang Kroutil. And before just jumping into the presentation, I would like to shortly introduce Wolfgang. So Wolfgang Kroutil is a professor for biocatalysis here at the University of Graz in Austria. He is holding numerous prestigious awards such as the Biocat Award, the Biotrans Award. He's a vice speaker of the field of excellence biohealth at the University of Graz, and he is a PI within the Austrian Center of Industrial Biotechnology. Wolfgang, the stage is yours, please.
Thank you very much, Chandra, for the kind introduction, and welcome everybody to this webinar. And it's my pleasure to be here invited and to present biocatalysis. And I try to entertain you, hopefully, for the next 45 minutes. Well, just to start and give you some ideas, if we speak about, we want sustainability, we want circular economy. What do we need for that? Well, there's always, we need, is one source as new additional source for the carbons, the renewables. But when we do the processes, we may use a bio-based solvent if we need a solvent. Well, or maybe if we can avoid the solvent, even better. But if we need a solvent, maybe we could use a water instead. And if we need for the process, we would like to use a catalyst, and there is where I step in, because I focus on the bio-based catalyst, because in the focus of my research there is the enzyme. We develop in the group biocatalytic in vitro synthetic methodologies, so not with living organisms, but like dealing like you do an organic chemical reaction. And we use that to do asymmetric synthesis or combine it. I will show you a few examples with chemo synthesis to do total synthesis to get some products. Well, enzymes are very useful to be combined together in cascades. I will show you examples there. Well, we're always interested in mechanisms and, of course, renewables. Over the years, we have investigated a number of enzymes. I'm not going through this long list. It just shows you that we have quite a lot of experience with different enzymes. I will mention some of those later. Well, you may ask, or why I'm so excited about the enzymes, or why do I think that an enzyme is a great catalyst? Well, you have here a fancy model of an enzyme. What you see here is the active site here, the entrance to the enzyme, to the active site. And you see here two substrates, one positioned in the center of the active site, and one is here bound actually to the enzyme. And actually, this is already one important aspect, I think, why the enzyme is a very useful catalyst, because it binds the substrate, the reagent, and not only binds that, it binds that in exact position to each other, in a very productive position to each other. So this exact binding to each other is one of the important aspects of an enzyme, and I think why it's useful. Because as a consequence out of that, it allows then to do a very selective, regioselective, chemoselective, and stereoselective performance for reactions. So this is due to this positioning. Additionally, due to this positioning, it allows that we do not need protecting groups in many cases. So this is maybe an additional advantage. What else is there? Well, you see here on top above the substrate in the active site some residues. Well, on the one hand, those residues are involved in binding, but those residues are also involved may be involved in activation of the substrate or the reagent. And we have not only one, two, but we have several in general around the active site. So we have this synergistic catalysis. Well, of course, there are also residues in the active site which stabilize the transition state. But I want just to emphasize, stabilizing transition state is just one aspect. We have those others, activation of reagent and exact positioning as other important aspects for an enzyme. There's one rather challenging part. Very often, substrate and product are rather similar. Well, we want that the substrate gets bound tightly to the enzyme, but of course the product should not be bound tightly. So it should easily be released. This is a challenge and not fully understood yet, but maybe one aspect which helps there is that the backbone of the enzyme is rather flexible. Normally we have this rigid structure, this picture of a rigid structure, but actually it's not perfect. Things are moving. Well, as the enzyme is built up by amino acids, and amino acids are chiral, we have a chiral environment. So that's why we have also the option for this high stereoselectivity. Well, due to the residues and to the many residues, we may have alternative mechanisms as with classical chemistry. Another aspect is a typical enzyme has maybe 300, 400 or even more amino acids. So I have 300 to 400 positions where I can tune the enzyme, so I can make variants. So there is a huge space of possible alternative catalysts. And finally, well, the enzyme is made up from renewables, so from amino acids, and therefore also degradable, biodegradable. So I think also this is a very interesting advantage. Maybe I show you one picture from the city of Graz, in the center of Graz, which is here the island of steel, which was a work of art from the year 2003, when Graz was the cultural capital of Europe. I think it's a model for an enzyme, because what you see here, it has a defined structure. It has in here, up there, you see a tunnel where people can walk in, and it has actually a cafe inside where you may get a drink, a beer, a coffee. So you may get activated there. So that's just what the active site is. You see here, actually, it's in aqueous solution. So that's mostly what we have in enzymes. And it is even attached to the background, so it is even immobilized here. So this is a rather good example here to tell in a very simple way what the enzyme is. On the other hand, as I already mentioned, this is of course not flexible. This is a stiff construction. But we should not forget, when we look at an enzyme, that actually the enzyme has not one conformation, but many conformations. So all that is actually a flexible structure, and we have quite a lot of movements there. Well, when we make the enzymes in our lab, so actually we make the enzymes ourselves, we don't buy them, but well, even when you buy it, it has to be made somehow. This is rather easy nowadays, in my opinion. So in general, we order the gene from online. So we order these 1,500 base pair sequence. In general, we order that already in a plasmid. Suitable in our case, mostly we use E. coli then. So we get the plasmid, so you get a tube of nothing, where you don't see a lot, but when you try and put that together then with E. coli, you can then start the expression. And I always say we produce our catalyst rather in a simple way. The student puts the E. coli with the plasmid, with the information for the enzyme, in a flask with salt and sugar and amino acids, and that let it shake overnight. the student goes home and hopefully sleeps well. And the next day when coming back, the first clear solution is then completely turbid. So the organism has grown, the E. coli has grown, has multiplied, and has then produced the enzyme. So it's just to filter off. And we then freeze dry the enzyme or the cells actually, and have then in general air and water stable catalyst powder, which is actually either the whole cells with the enzyme. So we break the cells and remove the cell wall and freeze dry then the cell-free extract. So we, in most cases, if not needed, we don't use pure enzyme because that's much to work. And that's also rather common when you order it. You will, in most cases, not get a pure enzyme because in most cases it's produced in E. coli, or it may be produced in another host, like in a fungus, like well, the old name Pichia pastori is well known. Then it may be excreted, then it has higher purity there. So we use those in our cases mostly freeze-dried cell power because it's simply easy to handle. On my first slides, I want to give you just a few reactions to give you an idea where in my opinion, biocatalysis is state of the art, and where it's also used rather broadly in industry. For instance, a reaction from our own work quite some years ago is the amination of a ketone. Well, this is formally it's a reductive amination, but as we use here a transaminase, and transaminases belong to the group of transferases, we use here isopropylamine as an amine donor as well as a reducing agent actually. Well, a recent example which we did in cooperation with Pavel Borovitsky is just to repeat to show the amination of acetophenone, which is rather easy, but also he then coupled that to produce an API. Well, very well known for this, so the transaminases are, to my knowledge, the enzymes in the meantime very broadly applied also in industry. One of the first examples was this sitagliptin, where this amino group on the stereogenic center was introduced by transaminases. And I think it was fascinating that actually here, an old process at MSD consisting of 5 steps, whereby one was making the enamine, another one was the asymmetric reduction, but just having 97% EE. Another step was removing of the rhodium, then a recrystallization to increase the EE, and finally crystallization to get the product, was substituted then by an enzymatic process, where in one step the enzyme introduced the amino group using the transaminase, just as I showed you before. And that was done with having right away the perfect EE. And well, we needed still needed the crystallization step to make the phosphate, the salt, but still the yield could be improved simply because you just needed 2 steps, which also led the overall process to less waste. And I think it was rather important for them to have here a biological degradable catalyst, and also they could use, they didn't need any special equipment to deal with hydrogen. So at the end, they could show this process or publish this process with a substrate concentration with close to 300 grams per liter in DMSO, which was rather impressive, but needed here definitely a lot of engineering of the enzyme. Another reaction, again, I start with, I show an example from their own work, and then is the reduction of ketones to alcohols, preferably done with 2-propanol as reducing agent, in general somewhere at 30 degrees in buffer. So the example is, again, in cooperation with Pavel Borowitsky from their own work, where a chloroketone was reduced and then transformed further, in that case, to dipropylene. But going away from those two very common, already used reactions, to show you a reaction which is also possible with enzymes. When we look to this product here, L-harmicine, which was isolated from this plant, and L-harmicine is known to have Leishmanian, anti-Leishmanian activity. When we looked here at this compound, we got intrigued that we may set up this 6-membered ring with the chiral center with the Pictet-Spengler reaction. And as tryptamine is commercially available, and also this special aldehyde was available, we found an enzyme which can catalyze indeed the Pictet-Spengler reaction, setting up this stereogenic center. So we don't need here any directing group, we don't need any chiral auxiliary. Or we actually never saw this product on the right side because that spontaneously cyclized right away to this precursor in optical pure form, which in a chemical step, we reduced the lactam to actually have then L-harmicine. And well, this actually is two-step synthesis and enzymatic a chemical step. So this is the shortest synthesis to harmicine as far as I know. I mentioned in my introduction already about cascades, so I just give here 2 cascades, and then later on I talk a bit longer about one example. Well, for the amination of alcohols, various types of cascades have been shown. So to transform here in my example, a primary alcohol, but we have also examples for secondary alcohols published actually just recently, to transform an alcohol to the corresponding amine, and that we have actually this hydrogen borrowing concept. So the hydride taking up in the first step, in the oxidation step to the here aldehyde, is then reused in the second step, so that overall we have a redox neutral amination of an alcohol. There are different other options for this cascade published by other groups like Nick Turner, or another cascade showing below, which I'm not aware that there is a chemical option for doing that. So having a phenol, which is in para position, not substituted, we could show that actually in a one-pot reaction with using 3 biocatalysts, we can introduce a vinyl group in para position, so doing vinylation there. So these is examples of cascade. Well, another cascade may be seen this dynamic kinetic resolution, where our aim rather recently was to show that we can also do a dynamic kinetic resolution of a secondary alcohol in water using enzymes only. So we used here an alcohol dehydrogenase actually to racemize the secondary alcohol, and an acyltransferase to enantioselective acylate one of the 2 enantiomers. I think at the moment there are quite some interesting reactions coming up from the area of biocatalysis. One is definitely CH oxidation. Well, for the oxidation of a CH to the corresponding alcohol, P450 enzymes have been long known, but they are more complicated to handle due to a rather sophisticated electron transfer chain. The new enzymes of significance, I would say, are peroxygenases, which can use hydrogen peroxide as the only reagent, giving as side product just water, and the 1 oxygen is incorporated in the substrate. You can also do, for instance, epoxidation. We use one type of such peroxygenases, for instance, to the alpha hydroxylation of fatty acid. I show here an example which we published recently to take octanoic acid and doing regio-selective alpha hydroxylation to give one enantiomer. Generally, the E is between 97 to 99, and we could show that actually with this variant which we designed, we could get rather high turnover numbers. Another reaction which we investigated was to do something like a Friedel-Crafts reaction. There are not too many examples with enzymes. Actually, we abused here or misused here an enzyme from nature, which there did a different reaction for the acylation of resorcinol to introduce here. And we engineered the enzyme that we can also have some bigger functional groups there. And very recently, PhD student Lila Gall in my group did investigated the formulation of resorcinol, and when we're using fluoroglucinol, we observed dye formulation, which is actually special to mention because chemically this is not easy to make the dye formulations, and the conversions with chemical methods were, to I admit to my surprise, lower than we achieved here with the enzymatic version. While the product can be transformed within one step to a molecule with antimalarial activity. So in the main part of my presentation, I would talk briefly about cascade transaminases, showing you an example of a cascade. Asymmetric oxidation of a CC bond, oxidative CC bond formation. Give you an example where we observed or exploited CC bond breakage, and at last amide bond formation. Well, in the first example, a natural product, dihydropyridine, actually that has antefidant activity against the pine weevil and some other interesting bugs. Well, we thought we can make this molecule if we break these CN bonds and have here a carbonyl. When you open on both positions, you would have a diketone, and well, you would need regio-selective amination and stereoselective amination, and only monoamination. So that was our vision, that we make monoamination in a stereoselective fashion and then getting the ring closure. And the enzyme, I mentioned it before already, is this transaminase. And we used here a one from Chromobacterium violaceum. What you see here, it's a dimer, and in the active site there is a PLB, pyridoxal phosphate, bound here to a lysine in the active site. Well, this enzyme taking, well, this and another one, we could access both enantiomers. So depending on the selected enzyme, and in that case, we used isopropylamine as a donor. We get the delta-1 pyridine, and we coupled it then right away in one pot with another enzyme, an imine reductase, to reduce the CN double bond. But in that case, we need a hydride, so we need recycling of the cofactor, which we did with 2-propanol. And you may say, okay, that's the sequence, but here we have an enzyme which is reducing, oxidizing alcohol to ketone groups, and my substrate has ketone groups in it. So I would expect side reactions that also there the ketone group may be reduced. And indeed, we saw these co-products, but these are all in equilibrium. So at the end, when you do it at 50 millimolar, we got of the desired single diastereomer at in 98% yield, both with perfect EE and DE, and the same was when we did it at 100%. And well, just to show you, we could do that on several gram scale, but at the end, we produced 5 grams of, for instance, of the RS enantiomer all together. The second example, oxidative CC bond formations. Looking at those two molecules, etoposide, teniposide, both are anti-cancer, but are semi-synthetic. You get them, extracting pothyllotoxin from the American Mayapple, and then within 2 steps, whereby one is an epimerization of the hydroxy, up there as well in the same step, demethylation, and then attaching the sugar moiety, you're at etoposide. Well, actually, nature makes this pothyllotoxin in a rather long sequence, starting actually from coniferyl alcohol, making yatein. So there is no hydroxy there yet. Then using an enzyme for this CC bond formation at the expense of molecular oxygen and chlorate. And then there is another step, an enzymatic hydroxylation. So maybe that one, this reaction here, the CC bond formation, caught our attention, and we thought maybe we can shortcut that. I will show you that we will use then a substrate where there is already the hydroxy there, because we could, that was easier to synthesize. Well, the chemical routes, there are several published. Here is one from 2016 to get to epipothyllotoxin in around 16 steps. But what we managed, Michael Fuchs supervised here, Mattia Lazzarotto, and they made first coupling these 2 molecules, whereby the brominated one was prepared in 2 steps before. Coupling those 2, getting here selectively this the diastereomer, of course in racemic form, and then coupling here this 3-methoxyphenyldoate, having this hydroxyyatein right away there. So this racemic hydroxyyatein, what you see here on the left, is actually really close to our etoposide, teniposide, except of the missing CC bond. It has already the hydroxy group installed. So we were wondering whether the natural enzyme could use this non-natural substrate and doing here the CC bond formation as well. And well, I would not have it on the slide, obviously, that it enantioselectively transformed the one enantiomer we wanted, making epipothyllotoxin. We could isolate that with 770 milligrams, which corresponds to close to 40%, but in optically pure form, could recover the other substrate. So we could get here to this precursor because, well, it has already the correct absolute configuration. So that in 2 steps it would be at the final product. We just need here, well, 1 enzymatic step, 2 chemical steps with 32% overall yield over the 3 steps. Well, the most limiting in yield is, of course, this last step, because we have a kinetic resolution on that. But still, it's a fast way to get there. Another example which we did was to look at salutaridine, which is on the one hand a precursor to thebaine or morphine, but also has various other functionalities of interest. And actually, you get from reticulin with an enzyme in 1 step to salutaridine. So we wanted to make this to this reaction. Looking again first at the biosynthesis. So the biosynthesis of salutaridine starts from L-tyrosine, and when we count all the steps, we are in total 10 steps, and until reticulin 9 steps. But what actually caught our attention was this intermediate, because nature is actually in that aspect, I think, here for this synthesis a bit complicated. It makes first the S reticulin and then inverts that via oxidation reduction to the R reticulin. So it consumes here quite some energy. So our idea was, why not make this achiral intermediate right away in a chemical fashion? And indeed, we did that by 5 chemical steps without, we didn't need protection of the phenolic group. So we worked out a method to get then this imine intermediate, which we could reduce in a stereoselective fashion to the R enantiomer with an imine reductase. And then, well, which is here on the one slide quickly shown, but in reality, it took quite some time to do the last step, namely to this coupling step. So that was the most difficult step, because when we use here the natural enzyme, actually, that is a membrane-bound enzyme, and to get to that to work in a, well, here we actually did a mixture of in vitro in vivo, but still we could get to the did in only or if we count 5, 6, 7 steps. So we were in total here in 7 steps, which is less steps than actually in the plant. So we should even shortcut what nature is achieving. Well, there were two examples for oxidative CC coupling. Well, oxidative CC cleavage, there we actually stepped in by coincidence. We thought to get to synthesize castorinin H, which is very similar to huperzine. The difference between these two molecules is the double bond here on top. And why did we not make huperzine right away? Well, this introduction of the double bond is the challenge. So we first, at the moment, looked to make castorinin. But we looked on how actually nature is making huperzine, as they are rather similar. And well, the biosynthesis for huperzine is the one described. And when we look at the last steps to get to huperzine A, which I zoom out on the next slide, then from this precursor, there are 3 oxidative reactions. The first is the introduction of a CC double bond, so a dehydrogenation. The second step is an interesting one to get out here, this CH2 and making the CC double bond. And the last one is an isomerization. Well, all these 3 steps, including the last one, also this is just an isomerization, is catalyzed by non-heme iron enzymes. So actually all are oxidative enzymes requiring alpha ketoglutarate as a co-substrate and molecular oxygen. And well, when you compare these last steps until huperzine C, so before isomerization, then you easily recognize that this is rather similar to castorinin. So it has the same pattern, except of this CC double bond there. So, and Stefan Bayer from my group has been to the group of Sabong in US, and there they made this N-desmethyl alpha obscurin. So then back in Graz, we investigated whether we can get actually from this N-desmethyl obscurin with enzymes to castorinin. And he showed that the first dehydrogenation step that works rather nicely. And he did it in a 1-pot cascade, but in a sequential fashion, meaning first he did the first step. When that was finished, he added the second enzyme and could then also indeed get this ring opening and get here this CC cleavage and get to the castorine. Well, starting from 100 milligrams over the 2 steps, we got 51% isolated yield. My last few minutes I want to use for amide formation, as that is in general a hot topic still, and just give you a quick overview about options how to use enzymes for amide formation and what we did there in the lab until now and what is published. Well, there are the options starting from the ester using a biocatalyst, starting from the acid as a second option. Well, we could start also from the nitrile, and well, there are some other very specific options, but well, in general, you would start from the acid or from the ester. When I look now, just starting from the ester, we have again two options, namely to be in an organic solvent. That is very well known, and I show you an industrial process, but there are many using lipases, esterases, so this is probably the most used enzymatic reaction. Or, which is not that most broadly used, to do this same in water, in buffer, in an aqueous solution. Because when you use here hydrolytic enzymes, there is a high chance that you will have hydrolysis of your esters to the acid. So you need to find a way that the amine is preferred, giving you the amide as the product. When we start from the acid, things get more challenging because, well, as in chemistry, there is activation needed. I will show you an example in an organic solvent using hydrolytic enzymes then later. So we could do it in an organic solvent. When we do it in buffer, we need activation. So as you would need in chemistry, and there are several methods there. There are, well, depends how you count, let's say here, 3 different options using all ATP. So these are carboxylic acid reductases, non-ribosomal peptide synthases, or ATP grasp enzymes, which were recently nicely shown in a paper from Merck as well. So those are all distinct enzymes with all very different substrate spectrum. And I think this is maybe important to stress. In biocatalysis, very often one tends to hope that there is a single method solving all the problems. That would be nice, but that's not or never the case. So we need for each type of substrate a different type of enzyme, and actually, well, even for the different, if we optimize, then the enzyme becomes more specific, but even then, in general, better. So there is never the universal enzyme solving the specific problems. This is a contradiction. So the more specific the catalyst needs to be, the smaller the substrate scope. Well, then there are other type of enzymes which are even more complicated, needing ATP and SCoA as a cofactor, and there's some. I just mentioned these 2, but I don't want to go into more detail for those. Well, actually, all 3 options, I keep it here as that. I show you on one slide just one process for the, well, esterification, amidation, actually, done on large scale. Start using an ester, making an amide inorganic solvent. So that is inorganic solvent in an organic solvent. So that is what is state of the art. Well, with that way we can make here a kinetic resolution for amines. I show you here now an example then from our own work where we then investigated more closely. When we look at APIs, then we have actually 2 bulky substituents on the amide. And that makes it for most lipases, when we started to investigate that, challenging. So that we have bulky, we consider that already here as bulky groups for a lipase. What at the end we found indeed an enzyme from Sphingomonas species, which we could use for the coupling of ethyl esters with various amines. So here shown just the scope for 6-membered rings. We always have an heteroatom in there. What helps if the reaction should work well is that the product precipitates. So that is actually the trick here. The product precipitates from aqueous solution. It works for many substrates. We've shown it for 5-membered rings, different substitutions, also for larger rings. In general, showing you for these two compounds here, at the beginning, the reaction is clear. So we're doing here at a gram scale, and within 20 minutes, it's stupid, and you just need to filter off the product to get then your amine in rather high purity in that fashion. So that is a rather efficient way to do that, starting from the ester in buffer. Another way where we make the proline amide as an intermediate for various drugs and pharmaceuticals. The idea was to get from proline and to substitute the current process for prolinamide making, starting from proline needing thionyl chloride and ammonia. The trouble here is that actually when you make the chloride, that racemizes, and well, we have 2 steps. There's another patent where catalysis is used with methanol, but over 2 steps. So this racemization step count was a point. At the end, we could use a candida-antarctica lipase variant, which was optimized in 2-methyl-2-butanol is solvent. Well, here these conditions are rather long, but still we have a slurry and have the amino acid suspended in the organic solvent. It's not dissolved at the concentration. Actually, the concentration is very low, but still we could make it in 1 single step. So this, to sum up, I hope I could show you that we can use enzymes efficiently in organic synthesis. That I showed you, hopefully that was my intention, some additional reactions to extend the repertoire of enzymes we could use. We misuse them all the time for non-natural reactions. And I think what is rather effective to design cascades to combine enzymes from different organisms and cut then short the synthesis. Well, of course, I need to acknowledge all the funding, well, especially, of course, all where the funding comes from PHARMECO, but also the Austrian Science Fund or European Commission and various companies, of course, cooperation partners. I would not be able to present here without my outstanding group. And of course, I acknowledge, as said already again, PHARMECO for giving me the opportunity to present the work here in this webinar. And I thank you very much for your attention here. Thank you very much.
Many thanks, Wolfgang, for the excellent presentation. I think we have 10 to 15 minutes for questions, comments. So I guess if someone would like to ask a question, he or she can either raise hand or we can consider also questions provided in the comments section. So while we are waiting for the audience, okay, some question already. So Vahat, please go ahead.
I think I have to grant access. Yeah. No, Vahak, you can open your mic, I think.
Okay, thanks.
Yeah.
Hi, I just had a question about dilutions using enzymes. That's usually part of the limitation for larger scale and production. how easy or fast these evolutions can be done or changes, or is it still a problem?
Well, it depends what is your starting point. You're absolutely right. When we do in academia and we develop new reactions, it's low concentration. We try to be in general at 10, 20, 50 millimolar. So if we are at 50, we are already higher. Well, you find, unfortunately, many publications also with even lower concentrations, which is connected, of course, to how good is it, how fast is it to evolve an enzyme. That question is not possible to answer in general.
Okay.
But well, it depends what facility it is to have. So if you have evolution capacities, well, for instance, the sitagliptin evolution, which I showed you, which was, I think it had, they introduced 27 variants over 11 rounds. As far as I know, let's say that took them 1.5 to 2 years. But you put quite a lot of manpower on that. So this is not a single person or not 2 person. That's to my experience, and that's still on it. I mean, we are even in the age of AI. We can predict a lot, but we cannot predict yet the best enzyme. That does not work. So definitely engineering is an important step. It's a it's a delaying step. If you start for reduction of ketones or amination, I think you have already, there are commercial libraries available already which contain very stable enzymes. So you would start there already at a very high level. So I would say at the stage of ketone reduction, substrate concentration should not be an issue anymore. Also for the amination of the ketones with transaminases. So it depends whether you have a good starting point.
Okay. So I was just hoping maybe that with the AI, maybe there's been some improvement, or maybe it's ongoing, that can be done quicker.
It's quicker, but not that quick in my opinion yet.
Okay.
It helps. It definitely helps. I mean, AlphaFold is one of the tools which is fantastic, but it helps to get ideas, but does not solve the problem right away. There is another question in the chat.
Exactly.
Thank you very much.
Thank you.
So Gabor Tasnaldi has a nice question in the comment section. So Gabor is asking, what about competing spontaneous hydrolysis in the case of the special lipase you presented at the end?
Yeah. Yes. Well, it's so indeed. Well, I did not go into detail, but maybe. So we have here, we are in aqueous solution, and we would have 2 reactions possible as we are using lipases. We would have the amide formation as shown, but as a side reaction, it's indeed the hydrolysis of the ester. The enzyme is very specially constructed. It prefers actually the amine because of a lipophilic site for the amine binding. So I have not the slides here ready, but in case asked questions, send me. You may send me otherwise an email. I can show you, give you more information on that. So it's the construction of the enzyme on the one hand, which makes the amine the preferred nucleophile over the amine. When you would leave it forever, well, very long, then the amide would get hydrolyzed as well. So you need to stop. That's why we left it shorter in general. would get hydrolyzed. But that's why what helps is well, that the product precipitates here, so that this follow-up step is significantly delayed. So when we mainly would see, well, when we take the amide, we would see hydrolysis of the amide. We did not spot that much the hydrolysis of the ester actually. When the amide is present.
So in the meantime, Gabor clarified his question a little bit. He mean non-enzymatic hydrolysis of ester.
No, that's no problem. No, that's negligible. No, that's negligible. That's negligible. That's clear. At pH 7.5, that is negligible.
Okay, so we have time for a few more questions, if there are any from the audience. If this is not the case, maybe I have one quick technical question. So, is it possible to perform enzymatic reactions neat without any solvents, and let me say even, for example, practicing mechanochemistry techniques, Wolfgang?
Sorry, the last sentence?
So is it any possibility or a chance to do enzymatic chemistry using mechanochemistry tools?
Well, you have mechanochemistry. There are examples with mechanochemistry. They may be extended, but it's easier to answer your first question. It is actually on an industrial scale, it's done neat without solvent. So I think it's, yes. For instance, the ester between myristic acid and the myristic alcohol for cosmetics is made in neat with a lipase. So indeed, well, as you could use the enzymes just in organic solvent, you could also use it in neat as well. So actually, from that point of view, it's not that surprising. On the other hand, yeah, it's exciting that you could avoid. One can avoid that. The examples with mechanochemistry are rare. There are a few examples out there. In some cases, well, probably, Shandor, you would not call it mechanochemistry, but yeah, well.
Yeah.
There might be.
I guess the definition should
room.
be kind of flexible here, because indeed, many processes, what we call today mechanochemistry, may not kind of, fall into the strict category of mechanochemistry in the reality.
Yeah, no, but there might be room for extension.
Oh, sounds good. So in the meantime, we have actually one more question in the comment section. So Tracy Cody asks, have any of these examples been examined under flow conditions?
So from the examples I've shown here, they have not been done with flow. So those exact examples, but for instance, but of course, so lipase reactions we have done also in flow. So lipases have been used in flow. Alcohol dehydrogenases, so ketoreductases, have been used in flow. Also transaminases have been shown in flow. So actually, I think, in my opinion, going to flow with enzymes is maybe even is definitely easier as for chemistry. Well, you need to immobilize the enzyme, and the conditions need to be compatible, but that is in most cases trial and error. So transaminases, alcohol dehydrogenases, lipases are commonly done commonly done in flow actually. Yes, thank you for the question. I hope that answers what was meant.
All right. So thanks a lot again for Wolfgang for the excellent presentation, and also we are grateful for the audience for the excellent questions. So yeah, with that I would like to close the event now, and yeah, thanks again for everyone participating. Have a nice afternoon.
Thank you very much. Thank you, Sandra. Thank you,
Thank
everybody,
you.
for attending. Thank you.
Thank
Bye-bye.
you. Bye.
Thank